Hi everyone. I see there are some people joining. Maybe you can wait like a couple minutes more if someone is still late, and then we'll start.
As we agreed, I am welcoming everyone that joined for this task force webinar centered in preclinical MRI, in which we will account with Irene Marco-Rius. The whole webinar goes around new MRI methods and preclinical models to assess brain tumor microenvironment. And first, Irene Marco-Rius will talk about carbon-13 hyperpolarization and metabolism studies. And then Martin Kristian Thomsen will talk about organoid models of human brain tumors for precision medicine. And then I will just start introducing Irene. It's my pleasure. More than a colleague, I consider her as a friend. And she is graduated in physics here in our university, so we are partially responsible for that. And then had a master in Heidelberg University in Germany. And after that, a PhD in biochemistry in Cambridge with nothing less than Kevin Brindle about hyperpolarized magnetic resonance technical studies for metabolism. So it's a great group. And after that, she has been in postdoctoral positions in California and also Cambridge in the United Kingdom. Always going around hyperpolarization combined with magnetic resonance tools to investigate metabolism, metabolic disorders, and to early treatment responses. And then in 2018, she returned to join IBEC, which is Institute for Bioengineering of Catalonia, as a Caixa Junior Leader Fellow. And then still going around hyperpolarization and magnetic resonance applications for real-time metabolic studies and organ-on-chip platforms. And then finally in 2021, now she's a group leader in the independent research group, which is Molecular Imaging for Precision Medicine Lab, still at IBEC, supported by La Caixa Chemical Biology Program. And then after that, she co-founded Vitala Technologies, which is a spin-off, bridging the gap between the lab, cutting-edge innovations, and the societal needs. And she has received numerous prestigious grants and awards, including Ramon y Cajal, which is an important grant, and after a FET open grant, and also ERC starting grant. So welcome, Irene, and thank you for accepting to participate in this webinar. We are looking forward. The floor is yours. Thank you.
Thank you for the introduction, Ana Paula. So as you've seen, my theme is hyperpolarization and carbon-13 NMR and MRI. So, now it's asking me to install something else. Hopefully this will work. You're seeing the full screen, right? I have some sound which might not play, and in that case, I will just skip the slide. So as I said, my whole career has been based on hyperpolarization, carbon-13 hyperpolarization, not so much on brain metabolism and brain imaging. So bear with me on that, but I hope that you will find what I'm going to tell you interesting nevertheless. So I just have a little outline on what I'm going to talk about. I'll start by just introducing this concept of hyperpolarization for those of you that may not be so familiar with it. Then I'll talk about how we're trying to use this technique with organ on chips and the planar in-chip technologies. And after that, I'll finish with some just ongoing developments at IBEC, just some case studies for you to see what we are doing. I always like to start with this slide. This is from work that was done by the group at UCSF in California. And what you're seeing here is a prostate of a man that had a tumor. And as you can see by the arrows in red, by doing conventional proton MRI, they could identify already where the location of the tumor was. And then what they did is that they injected a hyperpolarized substrate, in this case pyruvate, and they overlaid the spectra that they saw on the anatomic images. And by looking at the pyruvate to lactate conversion, what they identified is that there was another region that was not shown in the anatomic images, but that was being highlighted by the metabolic image. And they did a biopsy. They confirmed that on that side, the patient also had some cancerous cells growing. So with that, they already thought that this technique was powerful and that it could tell that the cell was already misbehaving or having abnormal metabolism before we could see the anatomic changes. And the reason why we can see that is the Warburg effect. So we know that in many types of cancer, when you give them pyruvate, there will be an increase in the lactate production compared to normal tissues. And that's what we are seeing here. So if we look at magnetic resonance spectroscopy, and I'm sure that many of you are experts, even more experts than I am on this. So we can do proton magnetic resonance spectroscopy instead of imaging. And one of the things we have to deal with is suppressing water. But then once we've done that, we can start seeing some metabolites and smaller signals that give information on, for example, metabolism, right? But we have very low signal-to-noise ratio, poor chemical dispersion. And then with that, it's also how we start using carbon-13 magnetic resonance spectroscopy, just pure spectroscopy, where we also see these metabolic peaks from the carbon-13 spectra. And now we have a better chemical dispersion. We still have an even lower signal-to-noise ratio. What we need to do now is that what we are injecting a substrate in many cases that is labeled with carbon-13 in strategic positions, depending on what we want to see, right? For example, here you would start with glucose, and depending on where you label it, you can follow the tracer. So imagine you label it in the positions with different colors, and you would be tracing it through the glycolytic pathway, for example, into TCA cycle, right? In the example I'm showing you here, they were giving 1.1 molar of glucose infused, and they had to acquire every 5 minutes. And in the end, they did an average over 40 minutes to see the type of spectrum that you see here in the back. So it's still very low SNR. And that's an intrinsic property that we have with an MRI, right? So we know that this is the formula by which we get monetization. So in the end, it's what gives us the signal that we see. And we can either modify the concentration of the substrate. That's why we inject carbon-13 substrates, for example. We can play a little bit with the magnetic moment, and we know that we will have more or less signal based on the nucleus that we are observing. And then we have that factor of polarization, which can also play a little bit around. So how do we increase sensitivity? Also, we can use different acquisition techniques. We can, for example, decoupling. We can have different hardware that would help us on that. So changing the field, the coils. We can increase the concentration, like we said, or we can go for polarization. How do we do that? Well, we can either go up in magnetic field or down in temperature. Down in temperature, if we're looking at biological systems, is difficult, but we can go up in the magnetic field. So we can do that by going up in the magnetic field. We can increase not only the signal sensitivity, but also the chemical shift dispersion, which is great. The negative or downside of that is that it's more expensive the higher you go on the magnetic field. In most cases, you need cryogens for that, and you need to have specialized facilities, which is not trivial. And again, if we're looking at humans or animals, then we have to take into account also things like peripheral nerve stimulation or energy deposition considerations that may not be so favorable as we're going high on the spectrum of the magnetic field. So what other solution do we have to increase polarization? Well, the technique of hyperpolarization. With that, we go to polarization levels of about 100% or nearly, right? In reality, we stay between 20 and 60%, but it's closer to unity than we can be by increasing field or lowering temperature. And in practice, what does that mean? So what I'm showing you here is a spectrum of W-label pyruvate, acquired 9.4 Tesla for over 2 hours, right? We have it in thermally polarized, so it was room temperature. And what we have is the energy distribution between the two states in the normal MRI conditions. When we hyperpolarize our sample, we are creating this population imbalance between the two energy states, right? And now what we will have is very strong signals, like you can see here. So instead of 2 hours, in less than 1 second, we get SNR levels of what we're seeing here, very low noise. And then in practice, I don't know here if you can hear my screen, but here what I'm showing you is what we are trying to do with hyperpolarization. Right? We are trying to get the signals that would be out of the noise. And we have a very limited amount of time to do that. So like in the example here, we only have a few seconds to a minute to identify the signal that we are detecting. So these metabolic signals, for example, and then the signal is gone. So we just have that very short time frame to really figure out what's going on in our biological system. And this can also be seen a little bit like in this image that I'm showing you here of the metabolism or the biochemical pathways that are in the cell, right? So what we can do is label specific substrates, for example, pyruvate, and then we can see going to the Warburg effect. We can play with substrates that talk about redox or cellular respiration or necrosis. And I'm showing you here the pathway by pathway because that's what we can do with hyperpolarization. So unlike conventional metabolism or metabolomics with NMR that you can see all these metabolites, with hyperpolarization we only see very specific metabolic pathways that are 1 or 2 enzymatic steps away, and also because of this limited amount of time. And this limited amount of time is dominated by the T1 relaxation. So there have been many substrates that have been polarized, for example, with dissolution DNP, which is one of the techniques that one can use. And you can see here what the relaxation time is for each of those substrates. So for glucose, for example, if we only have 10-second T1, that means we have to be really fast on the transfer from the machine to the injection to the acquisition, which limits what we can see. And I'll show you how the experiment is done so that you understand what I'm talking about. So this is a busy slide. What you will see is that David and Gergu, 2 researchers in the lab, are by the polarizer, so the machine that prepares the hyperpolarized substrate. They are here in this picture of the lab, so the HyperSense polarizer that we had back in the day. And you will see David walking across the lab to put a sample in the benchtop NMR spectrometer that we have at the other side of the room. What you will see here on the right is that the signal, with time, as the experiment progresses, will decay. So it's again dominated by this T1 relaxation. So let's start it. So you see here they are collecting the sample that has been already polarized. He's walking across the room. And in the sample, in the NMR tube, he has some cells. He's mixed with the pyruvate. The signal, as you see, nearly decay. We still have enough signal to be able to see this type of peaks where we see the pyruvate that we inject, the hyperpolarized substrate, which is injected at a higher concentration than the metabolites produced, and the buildup and then the decay, in this case, of lactate that was produced by the HeLa cells. And the focus of the research in the lab is precisely to work on technology development on one hand, and also we aim to characterize some of the biological studies that we are studying in the lab. So what are these models that we can study? Well, with hyperpolarization, there are studies already ranging from cell suspensions all the way to humans. So there are already clinical studies ongoing, especially in the US and the UK. And as you know, some models are better because they reproduce better the physiology that we want, but some others are stronger in that they are simple, right, to use in the lab. So they all have their advantage and disadvantage. Now, the organ-on-chip community has grown a lot, which are these multicellular structures that try to mimic in vivo physiology, a step in between in vitro and in vivo. And that's what we are targeting. We are aiming to develop the magnetic resonance tools to be able to study these models. And in case you're not familiar with these systems, they are microfluidic chips where you can grow your cells in a 3D structure so that they mimic or try to recapitulate some characteristics of a specific organ that has also some function. For example, a muscle that would contract, right? And IBEC is an institute that works a lot with organ on chips, develops models, sensors, And one of the things that we noticed when I started was that there's still a need for real-time in situ sensing of these cells that are growing in the chips. There's also a need for non-invasive or non-destructive readouts. And there wasn't a tool that you could validate both in vivo and in vitro. For example, with microscopy, you cannot put an animal, right, or a patient under the microscope and see the same thing. So obviously for that, I thought maybe hyperpolarization could help. And the idea would be that we could use the same technique for a human in the hospital, and then we could use that same technique and biomarkers in the chip, and then we could get information and do sort of a twin in these two models. So if we link it then to the whole workflow of what the lifetime metabolomics or following a sample throughout the whole lifetime of that sample would be that, for example, we could put an animal, in the future would be a patient in the MRI, we could do hyperpolarization or metabolic imaging on the animal. We could then take a biopsy or harvest the cells, make chips out of that, and then with the same technique, acquire data. And it's where we start working with disease modeling or talking about precision medicine in case that we can test some treatment and report back which treatment may work for the patient. And what I've shown you was spectroscopy, but we're talking about spectroscopic imaging. So now we want to localize spatially these MRI signals, right? Or hyperpolarized MRI signals. And that's what we do in the lab as well. As I said, again, with the focus of technology and animal model characterization. I'll just show you some preliminary data that we have on the brain because I know that that's what many of you are interested. So we again acquired just some anatomic images of a glioblastoma model that we have. And then we acquired a CSI with a carbon-13 hyperpolarized pyruvate, and we see the lactate production. And with that, we can do again the lactate to pyruvate ratio on each of the regions and try to compare what we see in the tumor with a region that has no tumor, for example. So this is something that we are also working at the moment in collaboration with VIO. And just to also mention it, I'm not going to talk about it in my presentation, but there's also another technique called deuterium metabolic imaging that you may have heard about. It's also metabolic imaging. We can also see metabolism happening, and there are many studies already in the brain. So I point to this one that was one of that revamped the field, and also this other one from Kevin, where they showed that different metabolic phenotypes have different treatment response by using this technique of deuterium metabolic imaging. So I encourage you to read their papers. And with this last part, I'm going to give you a very quick overview of things that we are doing at IBEC, going in the direction of combining hyperpolarized MR and organ-on-chip technologies. And it's a long step because, as you will see, there are many different elements that have to be all aligned and fulfilled. So when we do an organ-on-chip experiment in our lab, we always start by growing the cells, putting them in a scaffold or a biomaterial that in our case has to have high diffusion, putting it in a chip that we have developed in the lab as well, acquiring by NMR or MRI, and analyzing the data that we get after the hyperpolarized injection. And I'll show you first two examples more related to the biology side of the things that we do in the lab, one on biomaterials and the other one on a bioreactor for 3D cell models. And starting with the diffusion is that we noticed that we need very high diffusion or flow in of our hyperpolarized substrate into the scaffold so that it can reach the cells and be metabolized in the time frame of the experiment. And what you see here is one of the cryogels that we have identified that works very well for us. And you have the different images to see the different scale, how it looks with the pores inside. And we do it by freeze thawing this material, and then it creates these big pores. And it's very malleable. What you see here are two of the constructs, and you see that it's very squashy. If you remove the water, it shrinks a lot, so it's really full with water. So with cell media, if we put it in, and it's very easy to handle. So that's the material that we use, and it's the one that we use for this other experiments that I'm going to show you using a bioreactor. So what we did here is just a do-it-yourself bioreactor that we 3D printed all the parts. We tested the materials that would be biocompatible, and it was something that was not trivial either. And we tested what would happen in 2 configurations of the cells, in this case with HeLa, in 2D and in 3D. And in 3D, we tried to culture them for over 11 days, and we checked that they were still viable and metabolically active. And then we tested what would happen if we had that construct, the 3D cells in the scaffold, and we kept the flow on inside the bioreactor, or if we stopped the flow and kept it static for about 20 hours in the tube. And as expected, we saw that when it was static and there was no renewal of gas exchange and media, the cells were not so happy. We did hyperpolarized experiments in this tube, and this is the type of spectrum that we saw with the pyruvate that we injected, the lactate being built up and decreased, and we always see the pyruvate hydrate that comes from the pyruvate as well. And to show you some results of these experiments, when we did the hyperpolarized study by giving pyruvate and observing the lactate to pyruvate ratio for the HeLa cells, the metabolism didn't change that much. So when I say 2D, I'm talking about cells that were grown in the plate and then they were lifted right before the experiment. So they are cells in suspension in the tube. And when they are in 3D, they are in the sponge-like material. So they stay in this 3D configuration, in this biomaterial. And we repeated the experiment with Hep Z2 cells. And in this case, we did notice a big difference between the 2D and the 3D configuration. And we believe that it's because HeLa cells are very resilient, so they grow in nearly any condition, right? But with Hep Z2, they have a very clear preference to be adhered. So the moment we lifted them up, probably they started not being so comfortable with the environment. And then just to give you 2 last examples or case studies, more technical. So I'll show you what we've done with developing a benchtop NMR for lab on chip, and then how now we are moving to MRI applications as well for this type of systems. For the benchtop, I'm just going to be very brief. I'm going to show you that we've worked on developing a benchtop NMR spectrometer that we could have in the lab that would have a single sample detection, control delivery of the substrate, and it should be easy to use. That was our focus. It was a project in collaboration with Oxford Instruments, EDVABS, and Multiwave in the FED Open project that we finished about a year or 2 ago. And now we have the prototype. So what you see here is that we have a carrier where we insert the chip that you can see in there. We push it into the spectrometer that we modified for this purpose. And once it's inside, the chip is at 37 degrees. There would be the flow lines coming out so that we could inject, and the experiment is ready to start. So now we have that up and running. And the idea behind then moving to using the MRI is that we could do multi-sample detection. We still want to have controlled hypervised substrate delivery, and we were also aiming for lower technical variability. So by having more samples in one chip, we could potentially control and be more robust and more reproducible. And also the idea is that by having these 8 wells, and we control how we want the cell media or the flow to be distributed in each of the wells, we can also control what the conditions are in each of the wells. And what you see here is, for example, how we control that the distribution of hyperpolarized substrates reaches simultaneously in a controlled way to all the wells. And for that, we did the first proof of concept with pyruvic acid peroxidation. And what we did here was that we had buffer, a urea phantom that would not change and just buffer as well. So we were expecting to see the chemical reaction in the red wells. And that's what we saw. So we confirmed that the CSI acquisition was working well and that we could see the signal where we expected. And then with that, we could also move on to see differences in metabolism again with HeLa and Hep Z2 models, for example. We did always, we start with an anatomic image of the chips to see that we have everything in the right place. In this case, we tested some cell suspensions as well, as we see here, and we acquired the CSI. And with that, we convert it into the spectra, and we overlay it on each of the wells so that we can get metabolic information from each of the samples that we have. And now we can extract again information on the lactate to pyruvate ratio and start seeing if there are differences between the different cell models in this case. And another way of using it, now going to 8 samples in one chip, would be to then playing with the condition that we have in each of the chambers or the wells. Here we played with changing the cell media composition or the supplements that we were giving, or lysing the cells and not lysing them. And then we start seeing what differences in the signal that we observe are occurring. And with that, I think I reached the end. So I just want to say that the dream ahead, and that's something that we hope to see in many years ahead, right? Is, can this organ-on-chip really help reduce animal testing? So with our technique, we hope to be able to give information on metabolism in this real time and the time frame that we have, and also whether we can use this technique for personalized drug testing. So that's something that I will tell. And with that, I would like to thank my whole team, who has been working really hard on getting the data I showed you, and people that were in the lab before who also contributed, and all our collaborators and the funding agents. And obviously, thank you as well.
Thank you,
Great
Irene.
to hear to you, Irene. Thank you very much for accepting to be here today. And if someone was not in the start of the webinar, we will save the questions for the end. You can write it in the chat if you want, or raise your hand, but at the end, so we can just squeeze the 2 talks and let the questions for the end if you want.
Okay, so then let's move on. It's my pleasure to present Martin Kristian Thomsen for the next talk. Martin is a cancer biologist with a keen interest in vivo models as a tool to explore biological questions. He earned his PhD at the Institute of Cancer Research in London, where he studied prostate cancer. And following this, he conducted research at the Spanish National Center Research in Madrid, using genetically modified mouse models and patient samples to study liver diseases. And now, since 2017, Martin has led his own research group at the Faculty of Health at Aarhus University in Denmark. And the focus of the group is using CRISPR technology to study cancer in advanced animal model. And their work has resulted in many publications related to prostate, pancreas, brain, liver, and lung cancer. And today he will present his work on the preclinical models of glioblastoma that they have at his lab. So thank you, Martin. Go ahead.
Thank you very much, and thank you for inviting me. And I can tell you this is going to be a different talk from what you just heard, because I'm probably not really an expert on MRI. I should not say that, but I'm more an expert on animal models, and then we're using the scanning to actually follow our tumor progression. And that I will go into a little bit in this talk as well. I could just tell you for the interest that this is going to be on brain tumors, but we're working a lot with prostate as well. We've also done lung cancer and liver cancer, and for all those, we also use the MRI scans. So we're quite familiar with the technique, without that is me that's the expert. We're actually doing together with the hospital here at Aarhus. So let me see if I can move on. Actually, I should get a point as well, and then I will also move on here. And I can see, there we go. So short introduction about glioblastoma. I don't know how much you're aware of it, but it is an aggressive tumor we have in the brain. It's not very common. You can see the incidence is like 3 to 4 people out of 100,000. And it's hit like around when you are close to 60 years. The problem with glioblastoma is that we have very, very bad treatment of it, and it's very aggressive. So the median survival is 15 to 20 months. And we have like a treatment that is called the STOP protocol, but that's like actually surgery, irradiation, and then also chemotherapy. Or more or less it remains incurable. So it's something to definitely work on to see if we can improve this. there is many way of do classification of glioblastoma, and it's moving on over the time. So these things are changing as well. But what you can say is there is a discussion where it's arriving from, but some of them is coming from astrocytes in the brain. And then you have this classic one, or the prenormal, or you have the mesenchymal subtype. And the discussion is actually if you have like heterogeneity in the tumor anyway, so you see all the things. But for what we know over here on the mesenchymal subtype, many of them have NF1 mutations, and that's actually what we have been trying to model in our mouse models then. And what we're doing, and this is maybe familiar to you or maybe it's not, but I will go through it with a little bit of details because it's like a key thing for this, is that we are using our mice here. And for those, we can implant like tumor cells straight into the brain, or we can use adeno-associated virus particle to come with CRISPR guides. And those CRISPR guides you can pack together like in a construct, so you can have multiple guides. So we got guide 1, 2, and 3. And then we have a little thing here as well. We have a promoter for the GFAP, and GFAP is expressed by astrocytes. So when we have that, we can get expression of CRI. And this CRI here is a protein that can do recombination in the mouse genome if you make a transgene mouse that have what we call a lock-stop lock size. So it can remove this size here, and then you get expression of Cas9, which we need for how our CRISPR system to work. And at the same time, we also can get expression of GFP, so we can actually see what cells we have been hitting. So this is a mouse we're using, and we're actually using it for all our cancer studies. And then we're using our virus particle to come in, or alternatively, we're using cell lines, yeah? And CRISPR, very shortly, what you know is making mutation in the DNA, and it can either be repaired by homologous repair mechanism here, and then it can cut again, and then we get non-homologous end joining, and we get like insertion or deletion of some bases, and therefore we also disrupt the reading frame we have like in our gene of interest. So I have a little thing to go through here, and actually this is some mice we have been generating here where we had loss of RB1 or TP53 or P10, or over here instead NF1 and also P10 and P53. And for those, we actually had to derive some cell lines, and that's why the names are here. But this is actually like the primary tumor before the cell lines have been derived. And you can see the GFP signal here, so you can see where the tumor is developed, and you see it over here as well. And this is probably where you guys have a focus, because you can certainly see we can do scanning here with the MRI, and we can mark up where is the tumor, and in that way we can follow the progression. But it can also give us an idea about what type of tumor we get, so we can see if we see it more dense, or if we can see it more over here where it look a bit more diffuse as well. You can see if actually have been crossing the midline as well. And we're using this type of scanner here, that is the MEDISO medical imaging system, and it's actually only a 1 Tesla, and then we use the T2 scan to actually see the picture. And I saw before we had image from like a more powerful scanner. The image we have here is maybe not optimal for seeing a nice picture, but the good thing is that we have pretty quick scanning, and we can have quite a few mice coming through during the day, so we can actually follow our cohort of mice in that way. I should also say that this system also have the PET scanner on it, so we can also have a tracer on. I do not going to show you image for that today, but we are using different tracers as well. But what we have done was we have been generating some cell lines from some of these tumors, and you can see this is how they look when they're grown in vitro. They are actually growing as spheroids, so that will say they're in suspension. They look like small footballs that is floating around here, and they come up to a certain size, and then you have to split them and so on, so they still can keep maintaining. And you have a nice GFP picture of them here, so you can follow them. When we're working with CRISPR, we had to kind of validate also at our CRISPR system has worked, because it's not every single CRISPR guide is working in every single mouse. So there is a bit of a randomization in this here. And that you can do by sequencing, and you get some index sequencing out there showing if you have got an insertion or deletion in your reading frame. So you see that for the different genes here. But actually, it's a very nice if you can confirm it on protein level to see if you have loss of your protein. And you can appreciate the cell line we have here, we call AMD5. It has loss of P53 and P10. And then you can see some of the other ones have reduced expression. And in that way, it's showing also you get some heterogeneity here. These mice, you can, or these cell lines, we can inject back into the mice. And this is to show you how that can be done as well. And we can follow the tumor progression by the scanner here. So this is for the cell line, we scan like at day 21 and day 42 again. And we see the survival over here of the mice. So some of the cell lines are very aggressive and some of them have no, we can say actually they do not develop tumors. We still see the cells present in the mice, but they do not develop tumors. So there is a variation here between the cell lines. So this was a little of a sidestep into how you can use the cell lines. And we can, as I say, with the scanning image, you have them here. And cell lines quite often present a little bit more dense on the imaging when we're scanning them. And that can be nice for monitoring the size. And you can see like an early time point and a late time point. So there we say we could do a treatment study where we can follow how is the tumor progression. If we have enough mice, we can measure the size before and after treatment, and that way following it. And at endpoint, we of course can look at the GFP signal and the histology of it, and in that way evaluate how their treatment has been. But it's very nice for using the scanner to actually have an idea about both the tumor progression, but also for the animal welfare, because you can make sure that your tumor size is not getting too large. And therefore, we are still in, what can we say, treating our animal well, even though they're getting sick. I will tell you a little short story about a study we have ongoing and wrapping up at the moment. And it's gonna be a lot more focus on some of the mechanism we believe that is involved in tumor progression and how the tumor is forming. But what we have done here is we have been using our CRISPR system, and we have done that we have different CRISPR guides. So we have P10, P53, and RB1. And the other construct we have is that we have NF1 instead of RB1. And we can see huge difference in the survival of these mice. So with NF1, it's a way more aggressive tumor than what we see with the RB1. And what we also could see was that the RB1 tumor has a very dense tumor core, where we could see that when we had loss of NF1, we had a more diffuse tumor. And that we found quite interesting to follow up on, and we can see here on some of the histology and so on. But also over here, actually on our indel data, we can see we have a very high percentage of knockout over here. And that actually telling us that we have a lot of tumor cells in the biopsy, where over here we can see this has dropped down. And that's also showing we have a lot more normal cells there that we dilute out how much mutation frequency we're getting. So it's all going through that we have this diffuse tumor here. So what is actually driving this phenotype was one of the questions we asked when we moved on with this project here. And for that, we was doing a kinase activation screen to figure out if there was some kinase that was specific activated or downregulated between these two set of tumors. And this can be run on a type of chip that's looking like here. So it's like antibody based, but it's also based on that you have peptide and how they're getting phosphorylated. And what we could see was that we had like upregulation of this gene that's called ROC1. And ROC1 is actually known to be quite involved in cell migration. And we saw that when we had loss of the NF1. So that was in itself very interesting for us to find, and also something we could follow up on. We could do some validation with Western blotting and see that actually looked like this assay has been running to an okay level, so we could move on with it. As well, we had like exactly the same mice over here where we had done the cell line, where we had like with mutation of RB1 or the NF1. And here we have the cells I showed you from before, but we could see that they're growing slightly faster when we have mutation of NF1. They're also more aggressive. So that is maybe making sense in that way. And we could redo our kinase assay with these cell lines to see what we saw upregulated before. Is that tumor specific, or is it more like the bystander cells that you have in the tumor that actually is giving us this signal? But we could again see, we could validate that we could see ROC1 was upregulated in our tumor cells. Interestingly, we actually could see that the other ROC gene, the one that's called ROC2, was downregulated, and that shows that there is some differentiation between the two ROC genes in this situation. And why I'm mentioning this is because the ROC inhibitor actually has been used for glioblastoma, but has not been a success, but it's also targeting both ROC1 and ROC2, so it doesn't differentiate between them. So we want to use our CRISPR system, and this is to show you how you can, again, use the model here. So we could do a control construct where we just had P10 and P53. Then we could include our NF1 gene, as I showed you before. And then actually we could also include the ROC1. So certainly we're now knocking out 4 genes at once instead of the 3 genes before or the 2. And the advantage with this here is, of course, we don't have to intercross mice. So we are a lot faster with doing it in this way. And when you look at the survival curves over here, you can see that when we have loss of ROC1, we have a slightly increase in the survival comparing to when ROC1 is intact, when you see over here in the NF1. And you see up here in the control, P10 and P53 is developing a tumor very, very, very slowly comparing to. So that suggests that ROC1 is increased. Actually, if you knock out ROC1, it's increasing survival. So it's also say it's bad for progression. If we look at histology, we can actually see that we have a tendency to get a more dense tumor here when we have loss of ROC1 comparing to NF1. And this is data we have validated. We can see it over here in histology as well. And you can see here a more dense tumor mass here, which is coming up on the staining. Again, we have some interfrequencing over here to confirm actually that our system has been working. So this we could validate also with cell lines. So we could use our cell line as before, the AMG5 cell lines. And in that one, we could use CRISPR as well to knock out ROC1. And we have validated that over here by Western blotting to see that then the gene is gone. And then those cell lines we have been able to implant into the brain of the mice. And again, follow like how is the progression is going. And again, we see we have an increase in survival when we have loss of ROC1 comparing to when it's intact. And you see it here in the tumor size as well. And you can see here a nice little dense tumor comparing to what we have up here. So in that way, it's confirming the other data. And we carry on with these results here. So we were deciding to use single cell RNA sequencing to figure out what's behind this here. And I'm not knowing how much you guys are aware of single cell and how it's working, but what it really is that we can see over here the different groups. So in the green color, we actually have the tumor clusters, and then we have all subtypes of cells. So we have the microglia and the blue here and so on. But we are focusing on the tumor cluster. This is what we like. And what we can see over here, we can see Cas9 expression, and that's because we have the transgene mice where we actually get expression of Cas9. So it's a validation of also that we have the tumor cells there in this heat map. We were quite interested in this little tumor cluster here, and that's because we actually have a decrease of it when we had knocked out ROC1. So it looked like that we had like a smaller subgroup of the tumor cells that actually had changed the what can we say, the identity of what they were. At least there was a decrease in this cluster. And we have been trying to look into what it is actually is for type of cells that we have here. And what you have here is what we call the tumor cluster 2. So they have like a very distinct expression of a set of genes comparing to what we have in our tumor cluster 3 and in our tumor cluster 1 that we have over here. So, so that was something we could carry on and do some bioinformatics to figure out what's actually going on here. And I'm just going to show you what we have here. So these are the different, when we go in and do like Go analysis of what we have up and down regulated in it. And the interesting part is that what we have here in this tumor cluster 2 is actually we have migrating cells, but they are also non-dividing. So that we say cells that is migrating away from the tumor. And that can also be cells that is coming back to why we see this more diffuse phenotype when we have intact of ROC1, where comparing to when it's knocked down. where you can actually see that in the tumor cells, you have some cells that is dividing, and then you have some that is quite well adherent, but there are non-dividing cells as well. So this is quite showing the heterogeneity we actually have in this tumor when we're generating them, and also what's going on. We also have looked at different genes, and one gene that came up was this one that's called perostatin here. And perostatin is something we have been following up on, but what we can see is a signaling molecule that is secreted, and it's actually signaling to the macrophage and the microglia and the parasite in the brain. And therefore we think this could also be an interesting gene when we're talking about how the cells are migrating in the tumor. And this is one of my last slide here, just show you how these migrating cells are looking. So what you can see here, you can have a dense tumor core here, and then you can see these migrating cells out here. And what we especially see is we see that these tumor cells look like they're migrating here around the blood vessels, which is actually quite classic for glioblastoma, that you can see there, like it's called in between the space between where you have the endothelial cells here and the normal brain here, and you have the migration here. And actually also these cells that we are back to the tumor cluster 2 as we're talking about, they actually have a little bit of a phenotype like parasites, and parasites is also involved here when we have blood vessel formation. So we think this is showing that the cells are getting this identity, and that's probably a reason why they can migrate in this area as well. So with all this, I will actually just conclude for you guys and tell you where we're working on. So right now we're focusing on that we have seen that the loss here of NF1 is driving this invasive glioblastoma. We believe that ROC1 is quite active in this part here. Not that everything is solo down to ROC1, but it definitely has a driving force. We see we have this small cluster of tumor cells that seems to be the migrating cells, and we find that quite interesting. And we're also looking a lot into what phenotype they have with the parasiting. What we know is that we have been looking further into this perostatin, and we actually have data now that show that it's showing also a dense tumor. However, it looks like it's actually different from ROC1, but this is something we are right now finishing up on, and we're looking forward to this. So this is still non-published work, but it's done by a PhD student in my group that is finishing up. And then I would actually like to just thank my group and the people that's been helping, and especially for the single cell, which we have Lynn. And then the medical doctor we're working with the scanning, he's called Mikkel Vendelbo, and he's the one that is in charge of that. So with this, I think I will stop and see if there is some question.
Thank you, Martin. Really cool talk. So if anyone has any questions, you can either raise your hand or write it in the chat and we can read it.
Hi. So first, thank you for organizing and to the speakers for the very, very nice talks. Very different, as outlined by Martin, but very, very nice and very comprehensive. And I think addressing very well what the topic was for this webinar. So I have a question for Irene. I could have many questions. I will narrow it down to asking about the radio frequency coils, because the organ on the chip, microfluidics and different bioreactor approaches that you showed are very, very impressive. And I was wondering if you also design the RF coils or if they are commercial, if you acquire them. And what do you think would be the role of cryogenic coils in this field? I also have then a question for Martin if I have time, but for now, I'll leave it here.
So I'll give you a quick answer. So there are some coils that are already commercially available for carbon-13, and those are the ones that we use for the benchtop NMR for the bioreactor. We develop our own coils for the benchtop in-chip spectrometer. So for that one, you saw that the coil was embedded in the chip. We developed it. And for the MRI, the first approach that we've done is to use a volume coil. So our idea was that anyone who has an MRI, a preclinical MRI with carbon-13 capabilities and a volume coil can use our platform. So we wanted to make it as versatile as possible. It's not the best solution. We know that the volume coil for a chip doesn't give us the strongest SNR, right? So the next step will be to make our own coils for that specific setup. But then we know that it's not so easily distributed to other collaborators, for example, or anyone who wants to use it. So there's room to do research and improvement in all of those. And obviously, I mean, having cryo coils, you always improve SNR there dramatically. Then you have the trade-off again of cost. So are you willing to pay the money that it costs for the SNR that you get? And then how much do you need the platform to be translated or easily shared with the clinic or with other partners? But at a technological point of view, it would be amazing to have it, yes, for carbon as well.
Nice. Okay, thank you. And since I'm still live, could I ask a question place a question to Martin as well?
Yeah, of course.
Yeah, okay. I could also ask different questions. I will also stick to one. So by the end of your presentation, Martin, amazing talk. Again, congratulations. You spoke about the invasion, which was one of the take-home messages, and you were showing us how this invasion was along the vessels. And I was also wondering if you're also seeing invasion along the fibers, the white matter fibers, which is also well described. If to which extent are you seeing it? Is it more prevalent along the vessels as compared to that sort of invasion, the white matter?
No, it's a really good question, and actually is something we're looking into in very detail right now, because there's many different ways of invasion in these tumors. And I think it's not like that it's just only one way that the tumor cells are migrating. And we also see that there is both what we call white matter, but also gray matter. And it's a bit tricky to actually backtrack completely what has happened and what's going on. But we see that this invasion is happening in different ways. And I think it's it's something I have some discussion with clinicians about as well, because it's also how translational is it to the human brain. There is some missing links here as well between our model and what you see in patients. But I think in general, this is super important to delineate, and especially also to figure out what different mechanism there is behind, because of course, if you can target like the spread of the tumor cells, you will be in a lot better situation if you had to do a surgery resection of the tumor with a patient. So it's something we trying to nail down on, and I'm definitely not sure we're gonna manage to do it, but we hope to at least get a little puzzle sorted in that way.
Okay, thank you. Fully agree. So each model will model a specific aspect normally of the disease. Okay, thank you.
I was wondering which is the spatial resolution that we can get with this MRI based in carbon-13. We have, maybe we have already tried it in the past when we had the hyperpolarizer, and there was two large voxels. I think we were missing more detailed spatial resolution. We no longer have it, but I would be interested in know when you go around the tumor, because I think the idea is maybe to get to do it in vivo sometime. And which is the spatial resolution we can achieve with this?
So it's not as good as with Proton, that's for sure, because we are using sequences that are really fast acquisition of the whole field of view. So the best that we can get is in the range of 2 to 3 millimeters square, right? So that's not small. It's still, we use it for in vivo applications in the liver, for example. And there we are not going for small features. We now have a spin aligner, which gives us stronger polarization than the hypersense. So we reach now about 60% polarization. With the hypersense, you could reach about 20% polarization. So we are hoping that because we have stronger signal, also we may be able to improve upon that. But I think that that will be the constraint in general as well, given the pulse sequences that we have, the gradients and everything. So, but we could try it. So if you're willing, come anytime to IBEC and we can see what we can do.
Yes, the other thing is one thing that does not have solution as well in proton, where we have different cells, tumor cells, macrophage cells. All of them have different metabolism. So sometimes we don't really know where this specific metabolism is coming from. So it's a bit difficult to.
Yes, here we would see it as a group. Yeah,
it happens. Spectroscopy. So.
Yeah, exactly. Here we would have it as a tissue rather than a single cell type version. Yeah,
Thanks.
That's still a limitation.
I will come.
Yeah.
And so we are here, Martin. You showed quite slow growing tumors, but in comparison with tumors, I know that it's a totally different model. But you talked about 60 days, maybe 2 months of evolution in this tumor, or maybe I missed something.
No, we have two models. So if we have the cell line, so we see it quite fast. But everything is relative because it depends what the model you have. Actually, I would say 60 days for cell lines is not that fast. There's many cell lines where it's going a lot faster. And you can try to slow it down with injecting fewer cells, or you can increase the speed with more cells. Yeah. I think the problem with the cell line is that you already have something that's very aggressive. So it can be difficult to maybe modulate like an effect if you want to do a treatment effect, and your window of treatment is not as large as you could wish. So we are using a lot more our CRISPR model, and those have like a human endpoint after 100 days, more or less, depending on what genes you're targeting. Yeah. So in that way, you're working in a range that we think is more suitable. Yeah, you don't want it to take too long because you also have to get a progression in your research. Yeah, but you also need to have a tumor evolution going on. Yeah. So it's a trade-off comparing to what experiments you want to do and what you want to address.
No, it's not bad. Having more time or a larger time frame gives you some advantage. Quite remember in the past, our model was a bit different, but we had a lot of variability, and I don't know if you also see it. So we could have tumors growing faster and tumors maybe lasting 2, 3, 4 months. Do you see it, or it's reproducible?
Meaning, of course, there is variation from mouse to mouse, but I think we're talking if we're taking those that going down at around day 100, like the first one will maybe be around day 95, and the last one at day 110, 115. So I think it's a very okay expectation. And of course, because this is virus delivery, it's also how good are you in your operation, meaning it's 2 microliter you're injecting into the brain. So you can very fast get like maybe 10-15% different amount of delivery. And that will also have an implication on what's going on. There, the cell lines are a bit more robust. And therefore it can be quite useful. We're quite happy with one of the cell lines because it's actually making diffuse and invasive tumor, where many cell lines actually are more growing like a bulk of cells. And that's not that representative. Yeah. So there is some few cell lines that I think is developing quite nice tumors. But again, it's also what is most useful. And the nice thing with the cell line is you don't need a transgene mouse. Yeah, so you can buy home your mice and you can inject the cells, and it can be very efficient. And you can also modify them ex vivo if you want to target with CRISPR or overexpress a gene and so on. So I think in that way they can have some advantages. Yeah. So.
Super nice model. Thank you both.
Thank you both of you for your talks. They were really, really nice. And thank you everybody for attending. So
And
that's
see
all.
you soon.
Thank you. Bye bye.
Yeah, thank you. Bye bye.